Oh, good afternoon, everyone. Let's get started with the next presentation on Chapter 26, the phylogeny or phylogeny in the Tree of Life. This is looking at the modern way that we do taxonomy. There is a connection back to the taxonomies of Linnaeus, but as you'll discover, we actually have advanced significantly past the sort of taxonomic comparisons that Linnaeus and previous and taxonomists who followed in the footsteps of Linnaeus until around 20, 25 years ago, perhaps. I'm losing my train of thought, I'm sorry. The way in which traditionally taxonomy has been done has reflected the way Linnaeus did taxonomy, which was basically on the basis of morphological and taxonomic, or morphological and anatomic comparisons, looking at living organisms and looking at their external and internal physical features and making comparisons on that basis. We have advanced our technology. We have access now to protein sequences. We also have access to DNA sequences. And that means that we have other ways of examining relationships. And that has led to a rethink of how we go about doing the process of taxonomy. It's assigning organisms to groups. And the new name of the system is called cladistics and cladistic analysis. It still leads to drawing tree diagrams, but those tree diagrams are built on a different basis. And that's something that we're going to dig somewhat deeply into in this presentation, looking at what historically we've done, what we now do, and look at the way we do it so that you understand how we draw these phylogenetic trees. And then we'll look at some of the consequences of our understanding about, I guess we could say, the origin and evolution of life on the basis of tree diagrams. And as you'll discover, there has been a substantial rethink, most of which has gone on over the course of about the last 20 to 25 years. So during the time that I've been teaching this class, I've noticed a substantial change in the way we think about relationships. And that's reflected in what I'm about to tell you this afternoon. So let's take a look and let's see what we can learn about building tree diagrams, something that most people are aware of. By the way, the history of tree diagrams, while people think that Darwin was the first person to actually draw tree diagrams, and there is a very famous page in one of his notebooks where he drew a branch tree diagram and wrote next to it two words, I think, indicating that he thought that this was how you could reflect the relationships between organisms in some form of diagram arrangement. In fact, tree diagrams in rather primitive form, certainly not as elegant as the ones we draw today, have been around pre-Darwin, but Darwin was the first one to realize the significance of creating tree diagrams as a way of illustrating evolutionary relationships. Obviously, you could draw tree diagrams to show relationships, but unless you understood the evolutionary relationship between organisms, something which really did not take hold until after Darwin published On the Origin of Species, then tree diagrams would not be a reflection of evolutionary relationships. And that's been a big change in our use of these diagrams. So that's something that's really important for us to pay attention to. Anyway, onwards and upwards. So when we start looking at this idea that we call the tree of life, something that has really been an accepted idea, starting with Darwin, but probably not a really firm idea until the early 20th century, when people were really able to start fleshing out tree diagrams on the basis of relationships by more formal analysis. I mean, we had the accepted relationships that Linnaeus had established and the people who followed in Linnaeus's footsteps. And somewhat ironically, those relationships, which had been established not on the basis of evolutionary relationships, but simply because it was thought that this was the way you could organize a divinely created group of organisms. Linnaeus is famously quoted as saying, God creates, Linnaeus organizes. So these relationships were on the basis of trying to pigeonhole and group organisms together. It would be those very relationships that Linnaeus and his colleagues and those people who followed on from Linnaeus, that Darwin would take and say those relationships are more than just on the basis of morphology and anatomy. Those relationships exist because these organisms are related to each other in evolutionary history. Those relationships reflect the fact that they are descended from a common ancestor or from common ancestors, if we want to take a bigger picture. And so we have a system of groupings, which most of you are most likely familiar with, either from high school, certainly from Bio 1, you were introduced to the more recent groupings, what is referred to as systematics and taxonomy, where you have the three major domains, or the three-domain system, the bacteria, the archaea, and the eukarya. And then below that, you have the taxonomic system that Linnaeus would actually recognize, because he was the one who first developed it, starting with kingdoms and working all the way down to genus and species. And we'll take a look at that. And I'll show you a quick way of remembering the sequence starting with kingdoms, because there are a couple of mnemonics that most people know. There's one that I like to use, and I'll illustrate that on the diagram when we get there. So there has been a lot of work done on pigeonholing. I mean, that's what humans do. When we see a apparently disorganized group of something, we want to organize them in a more formal fashion. And so beginning with Linnaeus and following up with others, and then, as I said, about 25 years ago, establishing a brand new way of looking at relationships on the basis of molecular sequences, we continued this idea of pigeonholing organisms. The irony there is that one of the most confusing things about taxonomy and systematics is the fact that it keeps changing. It's never static. And I fully expect that it's going to continue in that fashion almost indefinitely, in spite of all the best efforts of everyone to try and come up with a final product. And the reason why is because other than the genus and species, and well, let's not even say genus, the species designation, none of the others actually exist in nature. And even as we've seen in previous chapters, even the species designation is somewhat blurry. The lines between species can be blurred very easily. And so when we try and create categories on whatever basis we do, we're really trying to artificially place organisms into those categories, because those categories are simply not recognized by organisms. And so not surprisingly, over time, we've had to constantly rethink how we do things. And when I introduced you to eukaryotes in chapter 28, so a couple of chapters from now, one of the things that I will draw your attention to is the way in which eukaryotes have been recategorized over the course of about the last 15 years or so. And it reflects a pattern that has been observed over the last 100 years or thereabouts in terms of how we go about doing taxonomy. And a lot of it has to do with personal preference, I suspect. But you'll see what I'm talking about when we get to that chapter. Anyway, let's continue moving so that we don't turn this into a really long presentation. That's something that I want to try and avoid. So phylogeny, which is a term that you've been introduced to previously, is the term that references the evolutionary history of an organism. So when we talk about phylogeny or some type of phylogenetic evaluation, what we're really talking about is thinking about the evolutionary origin of a single species, or perhaps even the evolutionary origin of a group of species, depending on which level we want to do the classification at. And basically, phylogeny allows us to think in terms of evolutionary history at all levels of the taxonomic system, or the systematic system of categories. And it really does then give us a handle on evolutionary history. I mean, that's the whole point of doing this. We group them together on the basis of similarities, but we're also commenting very, very strongly about the evolutionary origin. In fact, one of the golden rules of the new system is that you can't group organisms together on any other basis other than shared common evolutionary history, as you'll see. So most of you are familiar with the concept of the binomial name, or the binomial nomenclature as we normally refer to it. And that binomial name, it consists of the genus name and the species name. So we write it genus, species. Now notice how I've written it here. Genus is always capitalized, and species is always all lowercase. If I was able to, I would write this in italics, because the two names are either written in italics, which is the more formal way of doing it, or if you're unable to do it in italics, you underline it. So the binomial name is either italicized or underlined. Oops. And genus is always capitalized. And those are standard rules. And that's where we begin. And most binomial names are used to try and identify something about the species that's being named, or the genus that's being used. But particularly the species epithet, the second part of the binomial name, is often assigned as a way of identifying where the species was found, or in some cases to honor someone that's important to the individual who is doing the naming, which is the person or persons who first identified this species and wrote up the species description. They are, that individual or group of individuals gets the honor of also assigning the binomial name, or at least the species name. The genus name may already be fixed if the species belongs to a genus. So the only thing that you really have that you may be able to assign is the species name. Once we have those, and again, this is all based on the work of Linnaeus, then we have to make sure that we realize where that fits into the larger taxonomic scheme that Linnaeus is also responsible for. So you probably are familiar with this systematic scheme of labeling and identifying the position of organisms in the taxonomy. And what we should bear in mind is that we, at the bottom, oops, I don't remember which screen I'm writing on. At the bottom, we have the most exclusive, most inclusive. So the domain categories are the most inclusive. And at the top, we have the species, and that is the most specific. And it also should be apparent that these are nested categories. So species are nested inside a genus, genera are nested inside a family, families are nested inside an order, orders are nested inside classes, classes are nested inside phyla, phylum, phyla are nested inside kingdoms, and the kingdoms are then nested into the domains. The domains are the only categories that stand independently and are not actually nested into something else. We also need to recognize that at each level, it's possible to have subcategories. And one of the things I'm often asked when I introduce this is, why do we have these subcategories? And the answer is because it's an indication that we're not entirely sure whether a category is fully inclusive or whether the category needs to be broken up into smaller groupings that are independent. So one of the things that you may not recognize about our own species name, which most people recognize as Homo sapiens, is that in fact, a more correct way of identifying us is Homo sapiens subspecies sapiens. And the reason why is because we are not entirely sure that modern humans are exactly the same as early humans, because all we have as evidence of early humans is the fossil record, and that only preserves the skeletal remains. It doesn't preserve behaviors, it doesn't preserve any culture or any of the other things that are ephemeral, things that are not preserved in the fossil record. So in some way you might say this is a little bit about covering all the bases, or as some people would say, CYA. But it really does reflect that in some cases we are uncertain about whether a single category is warranted or whether we need to break that category up into smaller groupings, each of which would be assigned their own category. So rather than having a lot of independent individual categories, sometimes we'll say that one category contains subcategories. And this is not unusual. If you take a look at any phylogeny online, you're going to see that. You should also be aware of the fact that different taxonomists will assign different names and different locations depending on the basis by which they're doing their classifications. So if you were to compare two phylogenies from the internet, for instance, you may very well find that there are some similarities, but there are going to be some glaring differences as well. And that's because the two individuals who drew up those phylogenies based them on slightly different ways of viewing things. And that means that whenever we do one of these categorizations, we assign organisms to categories. We have to remember that for the most part, unless we are dealing with living organisms, and even then, this is also true, what we're really stating is a hypothesis. It is one explanation of what we observe. It is likely to be the best explanation that we can create at that point in time, which is what science is all about. But on the basis of better data in the future, that may have to change, which is the reason why, much to the chagrin, I guess, of instructors like myself who try and explain this to our students, periodically we have to say, well, everything's changed, or this is what you may see, but this is what you're going to see because things are changing. This is the most dynamic and the most non-static part of biology that you can imagine at this point in time. So I said I would give you a mnemonic that allows you to remember these in the sequence in which they lie. So the diagram shows it from a bottom-up arrangement. In the interest of using space, well, I'll write it off to the side and I'll go from bottom upwards as well. So my mnemonic is King, Philip, came over. Oh, great. Spain. So it only covers the Linnaean categories, so from kingdom down to species. But of course, the three domains are a more modern derivation. Those have been around only for about 15 years, firmly accepted. And so we don't really have a way of including them into the domain as far as the sentence structure is concerned. But you can see that the first letters of each word in the sentence reflects the particular taxonomic category. So king for kingdom, Philip for phylum, came for class, Spain for species. There are a number of different ones out there, but they're very similar. If you have a different one that works for you, fine. All I want you to do is to be able to remember the various taxonomic categories and be able to explain which is where, because as we go through the various kingdoms and the phyla within those kingdoms, and in some cases we'll get down to class and order, it's important for you to recognize what it is that we're discussing in each of the groups that we discuss in the future, because once we get past this chapter and we begin the bacteria chapter, which is the next chapter coming up, we're basically going to be going chapter by chapter, group by group in the taxonomy, looking at their characteristics, looking at their origins, looking at their modern features, looking at their evolutionarily historic features. And it's really important that you be able to recognize where in the taxonomic scheme we are talking as we go through the different groupings of organisms. So make sure that you have an understanding of the taxonomic hierarchy as part of your understanding of this chapter, if you haven't already begun to do so based on the taxonomy lab that you've just completed. So when we start thinking in terms of trying to create a link between the classification schemes that were used by Linnaeus and subsequent taxonomists and our modern understanding of phylogeny, evolutionary history, we rely on the branching trees. Now, the branching trees do make comments about evolutionary history. This has been true since Darwin started drawing them to illustrate how he viewed evolutionary relationships, and we've continued to do that. But the trees that we draw today are built on different bases than the trees of the past. And that's one of the reasons why we have to spend so much time talking about the way we do phylogeny today versus the way it may have been done traditionally. But the nature of the trees and what the branching within the trees actually represents has not changed. So let's take a look at the branching relationships within a typical tree, a generalized tree, to illustrate what this is all about. So here is a tree of some carnivores. The first thing to realize is that the branch point at the base of the tree down here represents the common ancestor of all the individuals on the tree. And in fact, under the new guidelines that we use for doing or for drawing these phylogenetic trees, that is one of the things that must be included in any tree diagram. Not only are all the organisms that are part of the group that you're illustrating in the tree diagram required to be there, their single common ancestor, because they must all trace their origins back to a single common ancestor, must also be present on the tree, and it must lie at what we refer to as the base of the tree. We say that the common ancestor is basal to all other members of this group represented by this tree diagram. So the common ancestor for all carnivores sits at this basal branch point, and all of the carnivores represented on this tree trace their origin evolutionarily back to that single common ancestor. So when we look at the location of the panther on this tree, the leopards, you'll notice that it comes directly from that branch point. So what we can also say is that of all the members of this group, the panther is the most basal. It is the one that first diverged from the common ancestor of this group. So we can say here that this is the basal species, and it was the first to diverge the common ancestor. All the other species represented on this tree diverged later. So on the other branch is represented the line that leads to all other members of the group. The panther has its own line branching from the common ancestor because it was the one, it represents the group, or at least the species, that first diverged from the common ancestor of all carnivores. Then we have another branch point, which represents the point of divergence between the Mustelidae, which are the weasels. So these are the weasels. And the Canidae, which are the dogs. So the weasels and the dogs, the Mustelidae and the Canidae, share a common ancestor. And so they share a common ancestor with the panthers, but the panthers diverge from that common ancestor as a separate group, and the remaining group that diverged from the common ancestor then becomes the common ancestor of both the Mustelidae and the Canidae, sitting here at position 1. So then we have one line diverging to give the Mustelidae, diverging from the common ancestor with the Canidae. And then we have the other branch leading to the Canidae, representing the other diverging group that leads to the Canidae. Then again, when we look at the tree, we notice that there is a common weasel ancestor to all of the weasels, and the badger and the otter are divergent species from that common ancestor, which sits here. Likewise, over here in the Canidae with the dogs, we notice that the coyote and the grey wolf also represent two species that branch from the common ancestor of all Canidae, and here's the branch point that represents that common ancestor. So each branch point represents the common ancestor of the two species that are on the two branches that diverge from that branch point. And ideally, when we create these trees, each branch point should represent a dichotomy, two branches. As you'll see, that's not necessarily always possible. We do have ways of getting around that, but it's not an ideal situation. It's considered to be a temporary situation. So how can we look at this? Well, we can say that all carnivores share a common ancestor. However, the panthers represent an early group to diverge from that common ancestor. So panthers share a common ancestor with all the other carnivores further back in time, represented by the fact that the branch point that leads to the panther is deeper in the nested tree than the other branch points. Then the Mustelidae, the weasels, and the Canidae, the dogs, also share a common ancestor. More recently than the shared common ancestor they both share with the panther. Then if you look at where the branch point for the coyote and the grey wolf occurs with respect to the branch point for the badger and the otter, you might think that there is a difference in the time at which the otter and the badger diverge from their common ancestor versus the time at which the coyote and the grey wolf diverge from their common ancestor. If that's the impression that you're getting, you're probably wrong. Because unless that is expressly stated, that is not one of the data points that you can take away from a phylogenetic tree. There are phylogenetic trees that do illustrate that. This is not one. I'll show you one that does illustrate the timing a little bit later. But by and large, most phylogenetic trees do not make a comment about when the divergence point occurred in geological time. It simply is commenting about the sequence of divergence. So in terms of the sequence of divergence, panthers diverge first, otters or weasels and dogs then diverge later, and then wolves and coyotes diverge even later after the divergence of Canidae from the shared common ancestor with Mustelidae. Now, if you have species that do share a common branch point, you can refer to them as sister species or sister groups. And they do share some common features as a consequence of sharing a common ancestor, but they will have their own unique features as well. So you have to look for common branch points. So is a panther and a badger sister species? No, because they don't share a common branch point. They do share a common ancestor, but it's a common ancestor to all carnivores, not a common ancestor that's shared specifically by panthers and badgers and no other groups within this phylogenetic tree. So there are some rules of the game that we need to be aware of in order to be able to extract data out of these phylogenetic trees. And we'll talk a bit more about those rules as we progress onwards. So the basis for these phylogenetic trees and the basis for establishing groups and relationships is now encapsulated in something that emerged roughly, I heard about it roughly 20 years ago for the first time as it started to appear in the literature. I'm not a taxonomist. I've had some training in taxonomy in some of my undergraduate classes, but I'm not a taxonomist, so I have not read deeply in the literature as a taxonomist. However, I am familiar with some of the literature, and I started to notice this reference to the phylo-code about 20 years ago. It started to turn up in the textbook around, I want to say about 12 to 15 years ago. And the phylo-code has now become established as the basis by which we do phylogenetic analysis or create phylogenetic relationships. And at the heart of it all is something called a clade, and that is why we refer to cladistic analysis as the way of building phylogenetic trees under the phylo-code. And the first rule of the phylo-code is that the only valid grouping that you can create, which is referred to as a monophyletic group, is that that group must contain all the descendants of the common ancestor and the common ancestor. You can't exclude some of the descendants. You can't exclude the common ancestor to all of the organisms in the group that you're trying to define under the phylotype. That is the golden rule. That is the yardstick against which everything is measured. So if that's what you're aiming for, if that's the gold standard that you're aiming for, is it possible to create groups that are not monophyletic? And the answer is yes. We've been doing this for a long time. And in some circumstances, we have, in traditional taxonomy, created non-monophyletic groups. And we have to recognize the potential for those groupings. So let's take a look at a phylogenetic tree again and get some more ideas about the tree. So here is the common ancestor, which I established that idea on the previous tree. Here is the branch point where lineages are divergent. So this is basically not telling you anything that I haven't already established from the previous tree. There is, however, one thing that I didn't point out on the previous tree, and it's this location where we've got 3 branch points. So the ideal, again, the gold standard is two branches from each branch point. Here we've got 3 branch points, which means that it should not exist. But the reason why it does exist is because we simply don't have enough information to determine which of the 3 groupings, and there's the term taxon simply means a group of individuals that share common features. So you can have a taxon at any level in the taxonomic hierarchy. So a taxon is a group of individuals that share common features. And so you can have taxons at any level. You could have a taxon at the kingdom, you can have a taxon at the family, you can have a taxon at the genus level. Obviously, you can't have a taxon at the species level because that's not a group, it's individuals. But what we don't have here is enough information to be able to identify which of the 3 groups, which of the 3 taxons at this branch point diverged first. Is it D? Is it E? Is it F? We don't know. So this is a temporary solution until such times as we do gather enough information to be able to decide which of these 3 groups should be basal and to the other 2, and therefore establish a pair of dichotomy branch points, I guess, is probably the best way to put this. I was trying to think of a term, but I couldn't twist around or get around that to think how it should be. So we want to create branch points that are dichotomies. And so we need more information to establish 2 branch points that are dichotomies. So for now, we temporarily have this polytomy, this branch point that contains more than 2 branches, until such times as we can clear up which of the 3 taxons is the most basal of the 3 and establish a dichotomous, there's the word I was looking for, a dichotomous branch point that identifies that basal divergence and then establishes the other 2 as 2 branches on a second dichotomous branch point on the second branch from that basal branch. So what we might wind up with is something like this. Assume that D branches off first and becomes this one. Then we would create another branch out here, and we would place E on it, and we would place F on it. So if we clear up this polytomy, this might be what we find. That's what we would aim for ultimately. Right now, we simply don't have enough information to be able to do that. So we allow for this temporary polytomy branch point. So this slide is kind of ironic in that I state very firmly here that phylogenetic trees do not show patterns of descent. They don't indicate when a species evolved in geologic time or how much genetic change has occurred since the branch point. But as you're going to see, you can draw phylogenetic trees that do show that, but you have to very specifically identify that that's what you're doing. And we do actually give specialized names to those types of phylogenetic trees. They still are phylogenetic trees, but we do actually give names to them. Now, I can tell you at this point, for a couple of editions of the textbook, a discussion of these specialized phylogenetic trees that do show genetic change or geologic time points was dropped from the book because people were very adamant that you can't draw phylogenetic trees that illustrate that material. What I've noticed is that recently, and this I've discovered from the literature, and now it's returned to the textbook again, is that people still use these trees and they still refer to them. So I guess that there's either been a rethinking on the part of the authors of the textbook, or perhaps there's been a rethinking in the phylo-code. I've not been able to establish which. But we do now recognize that these trees do exist. The textbook doesn't refer to them by their specialist names, but I do in this PowerPoint. So when we get to it, I'll show you. I think I've also established this final point. Yes, we do state that sister species do sit next to each other in a phylogenetic tree, but on the basis of the fact that they share a common branch point. So these would be sister taxons, and they share common ancestor, and therefore a common phylogeny. If we've got this, these two are not sister because they're from different branch points. So just because two species sit next to each other in a phylogenetic tree does not imply that they share common descent, because they have to share a common branch point to demonstrate that they do share a common ancestry. If they don't share a common branch point with the taxon adjacent to them in the tree, then they're not sister species, or they're not sister taxons. So these phylogenies are not just exercises in drawing trees and creating potential evolutionary hypotheses. These actually do serve useful purposes. So one of the things that happened back in the 1980s was that all the countries that were involved in commercial whaling were gathered together under the auspices of the UN and the International Whaling Committee, and a treaty was established that basically brought an end to all commercial whaling. However, the Japanese government protested this and said that whale meat was actually a delicacy as part of the Japanese cuisine and part of the Japanese diet. And they asked for a clause to be included in the international treaty that allowed the Japanese to continue carrying out what they referred to as scientific whaling, which is basically for a euphemism for being able to continue to capture whales, but of specific types, arguing that it was a way of monitoring the whale populations. And basically it put the treaty, as far as the Japanese whaling quote unquote industry is concerned, put off limits all species from the northern hemisphere and only allowed a limited number of species, particularly the minke whale, to be captured in small numbers from the southern hemisphere. Now, as part of the treaty, the Japanese were also required to allow inspectors to enter the fish markets in Tokyo and take samples of whale meat that was being sold to identify which whales were being taken as part of this scientific whaling process. So here is a phylogenetic tree that was first published in the journal Science by the AAAS, the American Association for the Advancement of Science, that is responsible for the publication of the journal. And here you can see how a phylogenetic tree can be used to look at relationships between organisms. So remember, the only legal species that can be taken as far as this phylogeny is concerned is minke whales from the southern hemisphere. So all of the whales, all the unknown meat that is taken, that is on this branch, is legal because it comes from minke populations in the southern hemisphere. But notice that there is northern hemisphere minke, there is humpbacks, and all humpbacks are off the table, both southern and northern. These cannot be taken. And fin whales can also not be taken, and yet some fin whales were taken. This clearly indicates that the Japanese were not living up to the terms of that treaty. They were taking whales that were specifically identified as not available under the scientific whaling clause. Now, this is independent of the sort of whaling that is carried out by native populations. So in Alaska, Siberia, a couple of other locations, the indigenous peoples are allowed to continue their whaling tradition because they go out and catch individual whales using traditional methods, which are very labor intensive and do not put the populations of whales at risk. And they do so for the purpose of actually eating the meat. There is some suspicion that the level of consumption of whale meat in Japan is not as high as the Japanese government presented it to be. This is, again, part of an ongoing, I'll use the word discussion, because it's probably the most polite term that I can use at this point, between the Japanese government and the International Whaling Committee. There are other countries, most notably Iceland, that would like to see some limited form of commercial whaling begin again, not just this limited type of whaling under the auspices of scientific whaling. But that remains to be seen. We do know that the population of whales that were specifically targeted by commercial whalers are starting to rebound, but because some species of whales do reproduce very, very slowly, it may be many years yet before the populations recover to the point where even some limited type of whaling may be permitted. But for now, the only type of whaling that is permitted is other than what indigenous people do, is this limited form of whaling that the Japanese are permitted to do. This is the reason why you have conservation groups going out and protesting and trying to interfere with the existing whalers, because this evidence suggests that they are not honoring the treaty which their government signed to limit which whales they take. So this is an example of where you can actually use a phylogenetic tree, not just as an intellectual exercise, but as an actual useful tool. And remember, one of my big things is that knowledge without application is just trivia. And so drawing phylogenetic trees for the sake of drawing them, well, that would be trivia, but here's a way of actually using it as a tool with very useful application. So it moves past that trivia idea to something that becomes a useful tool. So, time for our first break, and we'll put Zoom to the test here, I guess. So here is my concept check question, which I will answer when we come back from the break. Referring to the phylogenetic tree of carnivores, are leopards more closely related to badgers or wolves, or are they equally related to both species? Explain the answer. So let me see if I can pause the recording, and we will come back from that recording, from that pause, from the break, to answer the question. So I'll see you on the other side of the break with the answer to the question. So welcome back from your break. That seemed to work out well. I think I may have solved my problem with Zoom. We'll continue on and see what happens. Anyway, referring back to the phylogenetic tree of carnivores, are leopards more closely related to badgers or wolves, or are they equally related to both species? Explain the answer. So let's go back and take a look at that phylogeny for a moment. So here's the phylogeny. Here's leopards. Here are badgers and wolves. Are panthers more closely related to badgers or wolves, or are they equally related to both? Well, your initial glance at this might suggest that panthers are more closely related to badgers than to wolves, because the branch point that leads to badgers from this common ancestor through the common ancestor of Mustelidae to badgers appears to be closer to the base where panthers branch off than the branch point that leads to wolves from the common ancestor with weasels. But remember, the horizontal bars here are not making any comment about the time in geological, or the point in geological time that the branching is occurring. It's just stating that the branching occurs. And it's the depth of the branching into the nesting of the tree that is really of significance. So first level of branching, second level of branching, third level of branching. So these two in terms of branch point or depth within the nested tree are actually at the same level, even though the horizontal bars suggest that they may not be. They're actually at the same level. So these two share a common ancestor at this point, and in terms of the divergence point of grey wolves and badgers from each other, this is the branch point that they share in common. And so they share this level of common ancestry. We have to go even further back in time to find the leopard. So the leopard is actually equally related to both wolves and badgers, because badgers and wolves share a common ancestor more recently than either of them share with the leopards on the basis of the hierarchical branching pattern in this tree. So badgers and wolves are equally related to each other, and therefore equally related to leopards as a consequence of this, because of the points of divergence and the depths that those points of divergence have in this nested hierarchy. So hopefully that makes sense to you. Now, let's move on and talk about the next concept, which is talking about or looking at the type of evidence that we use for creating phylogenies. So the traditional evidence for establishing phylogenies, fire up the pen again, has been fossil evidence and morphological evidence. But we now have evidence, molecular evidence, most recent, and considered reliable. So why have I put that statement in here? Well, fossil evidence and morphological evidence is subject to subjective analysis. Do these bones look exactly the same? Are there subtle differences that I can identify that would suggest that they are different species? Morphologically, as we've seen in previous presentations, it is an error to think that morphology, external appearance, and similar external appearance implies that the individuals belong to the same species, nor can we assume that differences in morphology automatically mean that they're different species. So fossil evidence and morphological evidence, while being our classic evidence for creating phylogenies, are subject to some level of error. The good news is that by and large, the vast majority of phylogenies that have been built on the back of fossil evidence and morphological, and we could include their anatomical evidence, have stood the test in the face of molecular evidence. So why do we accept molecular evidence as being more reliable? Well, if we are looking for changes, like changes in the skeleton, or like changes in physical appearance or behavior, or whatever sort of changes might lead to speciation, ultimately, at the deepest level, those changes should be reflected as mutations in genes. They should be reflected at the molecular level, either in the proteins or in the DNA. So the initial molecular evidence was on the basis of comparisons of proteins. But if you recall from Bio 1, what you learned about the genetic code, you should recall that there is a level of redundancy built into the genetic code, specifically what is referred to as the wobble base, the third nucleotide in a codon, in a triplet, that three nucleotide triplet that makes up the codon. For a significant number of amino acids, there is more than one codon coding for that particular amino acid, and where the difference occurs is in that third nucleotide. So what really matters is that the first two nucleotides be the same. The third nucleotide could be one of four in many cases. So if you were to have a mutation that changed the third nucleotide, the so-called wobble base, that would not lead to a change in the amino acid sequence. We actually referred to that as a silent mutation. Hopefully you remember that from Bio 1. And so while protein sequence analysis can give us a window on phylogeny, something that is more accurate in terms of actual changes that occurred that gave rise to speciation, more accurate than fossil evidence and morphological evidence. The real story of a species phylogeny or a group of species phylogeny is written at the DNA level. And we've really only just started to get good access to DNA sequences with the completion of the Human Genome Project and the development of quick and easy ways of sequencing complete genomes. We can now start sequencing genes from multiple species in a wholesale fashion, something that used to take years to accomplish when we first started doing DNA sequencing. I remember when I started in the DNA sequencing biz, if we thought we could sequence several hundred nucleotides every couple of weeks, we thought we were doing well. Now we can do multiple millions of nucleotides in a day. If you were to pay for getting your genome sequenced, I've had mine done twice. These days, the price is coming down below $100, and it takes usually 2 to 3 days once they receive your DNA to actually complete the sequence, once they start processing the sequence. It's very, very quick. And it's probably going to get quicker still. That seems to be the situation. It's going to get cheaper. It's going to get quicker. We're looking at the potential in probably within the next 10 years or even earlier of being able to sequence complete genomes in a day or so and do it for under $50. And this is when your genome data becomes part of your medical record. And that is actually starting to be the case. I'm not going to delve into that any further in this class, but it is something to consider. So this is the reason why we consider molecular evidence to be the most reliable, because it really does have the story of a species or a taxon's phylogeny established or written in that genetic code, that genetic sequence. So one of the problems that we run into is this idea of homology versus analogy. We've talked about this briefly before, but this is where it really becomes a problem. Because morphology, and particularly homology within morphology, implies common ancestry. So if you think back to when we were talking about the evidence for evolution and we did some comparisons between mammalian forelimbs, the reason why those forelimbs are similar is because they are all derived from a common mammalian ancestral forelimb. And so all of those forelimbs are considered to be homologous because of that common ancestry. But sometimes what appears to be the product of homology can actually be the product of analogy, or as we described it previously, convergent evolution, where two totally unrelated organisms converge on a common solution to a particular problem, because there are only a limited number of solutions that can be developed by natural selection and evolution. And as a consequence, if you're trying to solve the same problem, you tend to converge on that common solution to that, or to one of the limited numbers of solutions to that common problem. And so not surprisingly, analogy turns up fairly frequently. We see the same thing at the molecular level too, in terms of amino acid sequences, but more specifically, DNA sequences, and being able to sort through homologous DNA sequences versus analogous DNA sequences is one of the new modern issues with taxonomy and phylogenetics when we start doing DNA comparisons. So the example that I've got quoted here is a bat's wing and a bird's wing. Are they homologous or are they analogous? Well, the problem here is it depends on how you're doing your analysis. So if you're thinking in terms of forelimbs, well, a bat and a bird are both terrestrial tetrapods. I was having a senior moment. So they're both terrestrial tetrapods, and all terrestrial tetrapods share a common feature: four limbs, two associated with the collarbone or the pectoral girdle, and two hind limbs associated with the pelvic girdle. And bats' wings and birds' wings, under those circumstances, are forelimbs attached to a pectoral girdle and are therefore homologous structures derived as modifications from a common terrestrial tetrapod limb form. And so terrestrial tetrapods include bats or mammals and reptiles. Those are the two big groups of terrestrial tetrapods. Bats are derived from a mammalian ancestor. Birds are derived from a reptilian ancestor. As I've mentioned before, birds are dinosaurs in every sense of the word, and therefore are part of the reptile clade, however you want to classify reptiles these days, which is another confusing part, which we'll get into later this semester. But notice what we're doing here now. If we say that bats are derived from a mammalian ancestor and birds are derived from a reptilian ancestor, well, we know that that reptiles and mammals do share a common ancestor, that's true, but bats evolved as mammals and birds evolved as members of the reptilian clade long after the ancestors of both clades diverged from their common ancestor. So under those circumstances, a bat's wing and a bird's wing represent convergence on a common solution, or to a potential solution to a common problem. How do you fly? Birds and bats arrived at slightly different solutions to the wing, but they nonetheless arrived at the wing as a solution to flying. And so under those circumstances, because they evolved independently after mammals and reptiles diverged from each other, these have to be analogies, not homologies. But I just finished telling you that there were homologies on the basis of being forelimbs. So you can tell that there is going to be some confusion here in separating homologies from analogies, because it really depends on the level at which you're doing the comparison. And so you can, on that basis, create a phylogeny that looks right on the basis of doing the analysis from one direction, but is actually wrong when you do the analysis from a different direction. This is one of the reasons why we are now looking at DNA sequences as a potential solution for this, where we're looking at a potential homology versus analogy issue. Typically, the DNA sequence will give us a solution. But again, we still have to be careful, because it is also possible to get homologies and analogies in DNA sequences, and trying to separate those two is even more tedious, although today we have methods of doing that. So here's another example of convergent evolution, the marsupial mole and the eutherian or placental mole, and they share common characteristics. But again, marsupials diverged very early as a group from the common ancestor of all mammals, and that divergence took place before the ancestor of the marsupial mole emerged from the common ancestor to all marsupials, and the ancestor to the placental mole emerged from the common ancestor of all placental mammals. And so these are, again, examples of analogy rather than homology, because while they share a common ancestry as mammals, as different groups of mammals, marsupials versus placental mammals, they do not share that. Marsupials share a common ancestor that is separate from the common ancestor of all placental mammals. They diverge from each other long before the common, as separate groups, long before the common ancestors of either of these two species emerge. So they have to be considered to be analogous under those circumstances. And again, are they homologous or analogous? This is the confusion. This is the question we're constantly asking ourselves. Where do we want to draw the line in all of this? So when we start looking at DNA sequences, here's where the confusion starts to occur. If we assume that this is the common ancestral sequences, and these are some of the changes that occur, then we wind up with these two apparently not homologous sequences. They have diverged from each other as a consequence of genetic changes that have occurred over time. So if we start with these, how do we know that these represent sequences that are derived from a common ancestral sequence? It's very difficult to do by visual comparison. The good news is we now live in the age of big data, meaning we have computer algorithms that can go in and search through all of this data. And because we now have rules that we can incorporate into those algorithms that describe what we now recognize are the limited number of ways in which DNA can actually mutate, there are only certain substitutions that are allowed. There are only certain types of deletions and insertions that are allowed, but specifically substitutions are the real key, because there are quote unquote rules of the road that govern the sorts of substitutions that can take place. We can now allow those algorithms to search through sequences that we're trying to decipher as homologs versus analogs and decide whether the sequences are in fact homologous or analogous. We also incorporate into the algorithms what are considered to be acceptable percentages of commonality that would cause us to say this is homologous versus analogous. Because remember again, sequences of nucleotides, as we define as genes, are somewhat random in nature. All that matters is that the sequence of nucleotides be interpretable as a sequence of amino acids that serves a particular function within the cells of the organism in which it's found. And so while it is possible for analogous sequences that share nucleotide similarity to occur in two different, totally unrelated species, there are levels of similarity that, on the basis of the rules that we understand, apply when mutations occur, that allow us to assign a level of similarity that reflects homology versus a level of similarity that reflects analogy. It is a far more complicated process than that. I have some grasp of it, but it is probably beyond the scope of what we need to get into in this class. All we need to remember is that big data analysis of gene sequences allows us to identify which gene sequences should be considered to be homologous versus which gene sequences should be considered to be analogous. So let's take a quick break. I'm not going to stop the recording at this point. I'm just going to continue on. But first, let's look at a concept check question and decide which of these combinations of features should be considered to be analogous and which should be considered to be homologous. And we should probably try and figure out why we're saying homologous versus analogous. So porcupine quills and cactus spines, they do look kind of similar. And if you didn't know anything about porcupines and cactus, other than that they have spines, there might be a temptation to say that these are homologous, they share some sort of common ancestor. And perhaps they do in very, very deep geological time. In fact, they must at some point in very, very deep geological time. But we do recognize that porcupines are mammals and the quills are the product of fusion of hair to create a rigid structure. So quills are actually modified hair structures, and cactus spines are actually modified leaves that have been severely modified to act as protective structures and also to reduce dehydration through transpiration. And so the reality is, as similar as quills and spines may look, they are in fact analogous, because both of these species do not share a common ancestor. So these are analogous. A cat's paw and a human hand. Well, this is a no-brainer, because this just takes us back to our first chapter, chapter 22, in comparison of forelimbs. They're both mammals. They both share a common ancestry of mammals. Their forelimb is a product of descent with modification from a common mammalian ancestor. So these have to be homologous. So that one's a bit of a no-brainer. An owl's wing and a hornet's wing. Well, again, it's a it's a solution to a common problem. How do you fly? But when you look at an owl's wing, that's actually a bony wing. It's a terrestrial tetrapod forelimb with the one big bone, two big bone, cluster of little bones, digit format. And then most of the space of the wing is empty space filled in with feathers to form a flying surface. In the case of a hornet's wing, it's a membranous wing. Its derivation is very, very different. Hornet is, of course, an insect. It's an invertebrate, comes from a very different part of the animal kingdom. Only in very, very deep time do owls and hornets share a common ancestor. So again, these must be analogous structures. So that's how we define these, and that's the basis by which we do so. So now we are going to begin looking at the actual process by which we use the combination of fossil data, morphological data, and especially molecular data at the DNA level to create phylogenetic trees. The process is called cladistics. The reason is why, or the reason why, is because the groups are referred to as clades. A clade is a group of organisms that are combined or are grouped together on the basis of shared common ancestry. The gold standard here is a monophyletic clade that actually meets that definition. All the individuals within the clade, all the individuals within the group share a common descent from a common ancestor, and that common ancestor is part of the clade that we are defining, or the group that we are defining. So a clade and a taxon are not equivalent. That's something to be very clear on. Can a clade and a taxon be the same thing? Can a clade actually be a taxon? Can a taxon actually represent a clade? Certainly, if the taxon meets the definition of a monophyletic clade, a group of organisms grouped together on the basis of shared common descent, including the common ancestor from which every individual in the clade is descended. Yes, a taxon can be a clade, but it is not implied that if you create a clade that it is a taxon, nor should it be implied that a taxon represents a clade. You can go backwards and forwards, but there is no implication that just because you've got one, you automatically have the other. That does not grow. One does not grow from the other, let's put it that way. So a monophyletic clade is the only valid clade under the rules of the phylo-code and cladistics. But there are two other circumstances that can occur, and both of these circumstances have occurred within the animal kingdom, as we will discover, not at this point in time, but well, we may touch on part of it today, but we'll certainly touch on it later when we do the animal kingdom later in the semester. So the two alternatives, well, first of all, here's a monophyletic clade, and you can see quite clearly that the clade as it is being defined here includes all the descendants that share a common ancestry, and also include the common ancestor at this branch point. So this is defined as a monophyletic clade. So when we take a look at this grouping, these individuals do all share a common ancestor. But the problem with this is that this group, G, also shares this common ancestor, but notice it is not included in the clade as it is defined by D, E, and F. G is specifically included. So the term we use for this is to say that it is paraphyletic, a parallel phylogeny or a parallel evolutionary history, implying that G shares a common ancestor with all the individuals within the group. And this has been the the situation that has existed for birds within the Linnaean system versus birds where we place them now. So Linnaeus identified birds as class Aves and reptiles as class Reptilia. But we now know that birds, as true dinosaurs, are part of the clade defined as the reptiles and must be included in there, because if you don't include the birds into the clade that is defined as the reptiles, then you've got a paraphyletic group, because birds share a common ancestor with all extant reptiles, going back to the origin of reptiles as a group. So to exclude birds and create a separate class outside of reptiles for birds creates a paraphyletic grouping. So we now consider birds and reptiles as a group, as a single group, and part of the problem now is trying to decide where we place them in the hierarchy. We may get into that when we do the animal kingdom. A polyphyletic group comes about when you have a lot of organisms that are grouped together that may share a common ancestor in the deep past, but do not share a common ancestor in the immediate past. All right, so the common ancestor of A, B, and C sits here. The common ancestor for D as part of this group sits here, which is not specifically included in the group, in the clade. So this clade is now considered to be a polyphyletic group, having several evolutionary histories. D has an evolutionary history that is separate from the common history of A, B, and C. And this is the reason why protists have been such a problem in taxonomy. You may now recognize that the Old Kingdom Protista that you may have encountered, perhaps in Bio 1, but certainly in any biology class that you may have taken in high school, no longer exists. And you'll discover, if you're not aware of that, you'll discover that this semester. The protists have undergone a huge re-examination over the last 15 years, and particularly in the last 7 or 8 years. And the reason why is because there is such a huge diversity of protists. I mean, they were originally considered to be unicellular eukaryotes, but as you'll discover, that definition very quickly collapsed as protists became a catch-all kingdom for anything that didn't fit as a prokaryote or didn't fit into the 3 big eukaryotic multicellular kingdoms. If you weren't a prokaryote and you weren't a fungus, you weren't a plant, you weren't an animal, then you automatically became a protist. And even if you just look at the unicellular members of the Old Kingdom Protista, where you have photosynthetic individuals, you have non-photosynthetic individuals, and you have some that are photosynthetic when it's appropriate and predatory when it's not. And so you actually wind up with a huge diversity of protists, and it's very difficult to establish a single common ancestor for all of them. They seem to be derived from a number of different ancestors, depending on which of the huge diversity of protists you're looking at. And so Kingdom Protista, as it was originally defined and as it developed, became a polyphyletic group, which is the reason why Kingdom Protista no longer exists. And out of that has emerged, depending again on who's doing the classifications, maybe 8 kingdoms, maybe up to 15 kingdoms, maybe fewer larger groups, depending on how you want to do the classifications. As you'll see when we do chapter 28, that is unfortunately part of the history of protists. Yeah, it's quite confusing. So yeah, the gold standard here is the monophyletic group. It must be a group of organisms that all share descent from a common ancestor. It must also include that common ancestor as part of the group. If you exclude members that share a common ancestry, or you share the common ancestor, you exclude the common ancestor of all members of the group, you either create a paraphyletic group or a polyphyletic group. They are unacceptable under the Phylo Code. Unfortunately, if you don't have enough information to sort this out, you may have to temporarily accept that. But with enough information, you should be able to regroup organisms into monophyletic clades. And fortunately, as you'll see when we get into the animal kingdom, where most of the work has been done, many of the old taxons actually do still hold as monophyletic clades, fortunately. Not always the case, as I've already started to point out, but by and large. But there's still a lot of this ongoing discussion. So here is another example of paraphyletic versus polyphyletic, and these are some reorganizations of the animal kingdom that have actually had to take place. So looking at the morphology of cetaceans, whales and dolphins and seals, you might think that they share a common ancestor, because after all, they share a similar morphology, they share many similar behaviors, they are air-breathing mammals, they live in the ocean, their body plans are modified for marine existence. So it would appear that they share a common ancestor, but the reality is that seals share a more close relationship to bears and dogs than they do to any other group of organisms. In fact, seals and bears are rather closely related, as are bears and dogs. I mean, we actually refer to the common ancestor of seals, bears, and dogs as a bear dog. It has characteristics of both. And we actually have a couple of fossils of bear dogs as ancestors of bears and dogs and seals. So if we group cetaceans and seals together on the basis of morphology, we've got a polyphyletic group because they do not share a common ancestry. Cetaceans, in fact, do trace their origin to a terrestrial ancestor, but that terrestrial ancestor, based on ankle bone analysis and on DNA sequence analysis, is very closely related to a group of animals known as Artiodactyla. These are the, these are the even-toed ungulates. The odd-toed ungulates are the I'm having a blank. I have trouble keeping track of some of these sometimes, and my mind has gone blank. If I think of it, I'll let you know. But surprisingly, on the basis of very close DNA sequence analysis, cetaceans' closest living terrestrial representative or relative is actually the hippopotamus. Figure that one out. You would not pick it. But if you look at one of the bones in the ankle, specifically the bone that allows the ankle to flex, there is a very specific feature, and I believe we looked at this in a previous presentation, that is uniquely found in all the artiodactyls and in the cetaceans, and not found anywhere else, which indicates that cetaceans are derived from an ancestor that is in common with the artiodactyls. And so when people want to group the cetaceans, one of the ways in which that is now accomplished is to create a nucleate called the cetartiodactyls. So the combination of the cetaceans and the artiodactyls, because that's where the cetaceans really belong. To exclude the cetaceans from the artiodactyls would create a paraphyletic group, which is what was the situation for a long period of time until the DNA evidence really confirmed what the fossil evidence had already indicated. So cetaceans really did share a common ancestor, common terrestrial ancestor with all artiodactyls. And so correctly, many taxonomists now have created this new clade, the cetatiodactyls, or cetatiodactyls, to group the cetaceans to their closest living relatives, the artiodactyls. So that's another situation where grouping organisms together on the basis of phylogeny on the basis of morphology leads to clades that cannot be supported at the molecular level. One of the other problems that we do have in establishing phylogenies, and particularly establishing cladistic trees or phylogenetic trees, is these two ideas: shared primitive, or as the book currently refers to them as shared ancestral characteristics, and the other group, shared derived characteristics. So a shared primitive or ancestral characteristic is a homologous feature that is not only found in the clade as you're defining it, but is found in other groups that are really closely related to the clade, but are not actually part of the clade. In other words, this characteristic predates the emergence of the clade from the common ancestor to the other closely related clades or groups that are not actually part of the clade. And so it is a characteristic that is more primitive, it appears earlier, than the derivation or the divergence of the clade as we are defining it. A shared derived characteristic only appears in the clade after the clade diverges from its common ancestor. In other words, it's uniquely found within all individuals that are part of the clade. And it is not found outside of, in groups that lie outside of the clade. So this is why we use the term evolutionary novelty. We use it very cautiously, however, because remember, very rarely does evolution create something that is truly novel. Generally, what it does is modify some existing structure to a new use. But sometimes evolution surprises us and does really come up with some uniquely novel features, and they can very definitely be used to define clades. So it becomes very difficult at times to recognize what is meant by a shared primitive or ancestral characteristic versus a shared derived characteristic. But the easiest way to remember it is that anything that is ancestral or anything that is primitive is found not only within the clade, but is also found in organisms that are closely related to the clade, but are actually part of the clade. Whereas a derived characteristic is always found within individuals that are part of the clade, but not found in individuals that lie outside of the clade. So hopefully that makes sense to you. I can tell you now that from my experience that historically, these are ideas that have created a great amount of confusion amongst biology 2 students. So I would strongly encourage you to take a close look at these ideas and make sure that you clearly understand what is meant by a primitive character versus a derived character. So I'm going to put a note here. I'm going to try and put it in slightly simpler terms. Yes. So hopefully that's a little bit of a simpler explanation of what we mean by primitive versus derived. It is a source of great confusion. And I've noticed that historically students have struggled to identify the difference between primitive and derived characters. So hopefully you're able to make that distinction. So the way that we can identify the derived characteristics versus the primitive characteristics is by using what you might refer to as an internal control when you do the cladistic analysis. If you think in terms of the cladistic analysis as an experimental approach, which it kind of is, and you recall from biology 1 that within any good science experiment, we include a control, a group that gets treated like the experimental group, with the exception of the actual treatment that is involved in the experiment. So all other conditions are kept the same. Then you could consider the out group to be the quote unquote experimental group in cladistic analysis. It's the yardstick against which you are going to measure those individuals that you are assigning to the clade to identify what is a primitive characteristic, which you would expect to be found not only within the clade, but also in the out group, versus those characteristics that are derived, which you would expect to find in individuals that are in the clade, but not found in the out group. So choosing an out group is very important. We want to choose a group that is very, very closely related to the clade as we're defining it, but actually lacks the very specific feature or features that define the clade. Will we find primitive features in the out group and the clade? Almost certainly. If they're closely related, we would expect to find primitive features, but it's the fact that those primitive features exist that allow us to use the out group as the out group. So I'm going to put a footnote here. Think of the out group as the control stick. Just so that we can, yeah, I'll put in here. So hopefully that last part is obvious based on what I've already said, but I'm going to just restate it again. So any features that are shared between the outgroup and the clade as you're defining it must be shared primitive features. Any features that are unique to the clade as you're defining it must be shared derived features. So let's take a look at an example of how we do this. And what I'm also going to do as we go through this is show you how we redefine the branch points as we do cladistic analysis versus what we used to do with taxonomy. So this is looking at vertebrates. It is defining the clade vertebrates. So what you need to know about vertebrates is that vertebrates are actually a subphylum within a larger phylum called Chordata. And there are, in fact, 3 subphyla within the phylum Chordata, 2 of which happen to be invertebrates. They lack the vertebral column that is the defining feature of all vertebrates. One of those 2 invertebrate phyla is called Cephalochordata, and the common name for the individuals that lie within Cephalochordata is lancelet. So here you can see a lancelet. Push that to the side a little bit. So lancelets, these are Cephalochordata. They are invertebrate chordates. So if vertebrates are chordates and Cephalochordata are chordates, as chordates, they must share some common features, and these would be the shared primitive features. And we define those as the 4 shared chordate features. And you'll learn more about these later in the semester, but they are a hollow dorsal nerve cord, a pharyngeal slits. Yeah, I'm having, for some reason, my brain is not functioning particularly well at the moment. So a hollow dorsal nerve cord, pharyngeal slits, a vertebral column, a notochord, not a vertebral column, a notochord, which is a precursor to the vertebral column, but it's a cartilaginous rod that runs underneath the nerve cord, and then a postanal tail, a tail that extends past the anus where the digestive tract exits from the body. So just to reiterate again, pharyngeal slits, which eventually evolve into gills, but then for non-marine chordates and, well, vertebrates, those tissues have been repurposed to other functions, some of which you've already encountered but don't realize it yet. But we'll talk more about that when we get to the end of the semester and do the animal kingdom. So pharyngeal slits, the notochord, the cartilaginous rod that eventually gets displaced by bone and becomes the vertebral column, a hollow dorsal nerve cord. So the nerve cord is just below the dorsal surface or the back, and it's a hollow bundle of cells rather than a solid bundle of cells. And then a tail that extends past the point at which the digestive tract exits from the body, otherwise known as a postanal tail. Those are shared primitive characteristics of all chordates. So the defining feature of all vertebrates is the vertebral column. And so you see the vertebral column here, but this branch point that represents the branch in chordates from the common chordate ancestor shared by cephalochordata and vertebrates is defined not by similarities, but be defined by the difference, the presence or absence of a vertebral column. So branch points are defined by differences, not similarities. And this is an important observation because in taxonomy, branch points represent similarities. Remember, the whole point of drawing taxonomic trees in the past has been to establish shared common features as a product of shared common ancestry. That's what taxonomic trees were all about. That's what the very first phylogenetic trees were all about. But now that we're drawing them on the basis of cladistic analysis, we're not using similarities to define the branch points. We're using differences to define the branch points. So the first branch point in this tree is defined by the presence or absence of the vertebral column. The absence of a vertebral column also establishes the lancelets as our outgroup, because all members of the clade vertebrate or vertebrata possess a vertebral column. So that's the defining feature of everybody on this branch and establishes the lancelets as the outgroup, which helps us define the vertebrates. Then amongst the vertebrates, there are a small number of vertebrates, the lampreys being the group that we're focusing on here, that lack jaws. And we refer to them as jawless vertebrates. In the fossil record, there's a large diversity of jawless vertebrates that predate and in some cases parallel the appearance of jawed vertebrates. So another defining feature of the vertebrates, as we know them today, is the presence or absence of a jaw. So the jaw occurs here, this branch point. Actually, let me do that in a different color. So this branch point is defined by the presence or absence of jaws. So the lamprey, being a jawed vertebrate, is still a vertebrate, but it lacks jaws. So this branch point now is defined by the presence or absence of jaws. And we can continue on through this whole phylogenetic tree, and I'm not going to do that because I think you've got the idea what the pattern is here. Each one of these branch points is defined by a difference, the presence of some feature or the absence of some feature. And so when it comes to tetrapods, yes, the tetrapod condition begins to appear in fish, but if we're talking about tetrapod limbs, those are found in terrestrial tetrapods, not in aquatic tetrapods, fish. So we have another branch point, the presence of walking legs versus the absence of walking legs. Then when we look at terrestrial tetrapods, we have those that have amniotic eggs. If you're not familiar with that term, you'll find out more about that when we do chapter 47. But suffice to say that amphibians lack amniotic eggs, reptiles and mammals, as descendants of a common amniotic ancestor, both have amniotic eggs. The big difference, of course, is that turtles, the reptiles lay eggs in shells. Mammals retain the eggs inside the body of the mother, the female. but they go through similar embryological developmental stages in both locations, and they possess what are known as the four amniote extraembryonic membranes. And we'll look at those when we get to chapter 47. Then if we want to define mammals as a group, hair is a defining feature because all mammals have it, whether it is obvious or not. Even cetaceans still retain a few hairs. If you take a look at the snout, the rostrum of a dolphin, you can find a few residual hairs still on the rostrum, on the snout. And the same with whales. So as mammals, they retain a few hairs, but hairs would be a disadvantage if you want us to move swiftly through a very viscous medium like water. So they have lost the vast majority of hairs. They still retain a few residuals or vestiges of the hair of their terrestrial ancestors. So hair would be a defining feature for all mammals. And so you can see how we can create phylogenetic trees by focusing not on similarities, but by focusing on differences. What are the derived features? Those are the features that establish the difference between the clade as we're defining it and individuals that lie outside of the clade. Primitive features are shared by individuals that lie outside of the clade with those individuals that lie inside the clade. So if we're defining mammals, a primitive feature would be the amniotic egg, because that's shared with all amniotes, including all the reptiles, which includes the birds, and which includes mammals. But it doesn't define mammals. It's a primitive feature. And so we can go backwards through this tree as well and look at that as an idea as well. But I hope this this explanation helps you understand how we do a cladistic analysis. We're looking at differences, and the branch points are defined by those differences, not by the similarities, as would have been the case in the previous taxonomic methods based on morphology and anatomy. We are truly looking for differences now, not similarities. So I mentioned earlier that there are phylogenetic trees that do comment on the number of genetic changes that have occurred post divergence or branch point, and also comment on the point in geological time when the divergences occurred. So the book refers to these still as phylogenetic trees, but people who use these on a regular basis actually assign names to these, and these names are fairly historic. They go back a ways to when we first started drawing phylogenetic trees. So phylograms, not phylogenetic trees, but phylograms are phylogenetic trees that diagram the amount of genetic change that has occurred post-divergence point or post-branch point. Ultrameric trees are phylogenetic trees that comment or illustrate the point in geological time when the divergent point occurred. So let's take a look at some examples of both of these by looking at trees relating to the same organism, same types of organisms. So here's a phylogram of vertebrates, chordates, and this is based on a protein sequence or amino acid sequence analysis of a protein that's found in common amongst chordates, but it's not found in Drosophila. So Drosophila here is serving as the outgroup to define the chordate group on the basis of this amino acid analysis. In this case, the line, the length of the horizontal line is a reflection of the number of genetic changes that have occurred since the divergence point occurred between, for instance, the lancelet and the common ancestors of the rest of the clade. We do have a long horizontal line for Drosophila because even though Drosophila is still extant, they're a living species, genetic change has occurred since the divergence between the common ancestor of Drosophila as insects and the common ancestor of chordates as chordates. So we can reflect the genetic change that has been ongoing in Drosophila as well. So when we look at the horizontal lines at each point, remember that is telling us where there has been or how much genetic change has occurred. So not surprisingly, because humans and mice are relatively closely related within chordates and certainly within mammals, because they're both mammals, only a limited amount of genetic change has occurred. And yet the line on humans is a little bit longer than the line on the mice. So that indicates that humans have undergone a little bit more genetic change than mice have undergone since we shared a common ancestor. That's the important thing to remember here. The position of the organisms with respect to each other is not necessarily important. What is important is the length of the horizontal lines to each of the organisms post-branch point. Let's take a look at an ultrameric tree. So one of the things that is important that separates an ultrameric tree from many phylogenetic trees is that all of the branch points for species that are extant must end at the same point in time. So all of these species are extant. In other words, they're alive right now. They're living organisms. So the branch points have to end at present. If they came to an end prior to present, that's an indication that the organisms that are being addressed or being diagrammed in the phylogenetic tree are extinct, no longer extant. So again, the horizontal line now from the branch point indicates where in time the divergence point is thought to exist, either in the fossil record or on the basis of other analysis, which we'll talk about shortly. So this time, the horizontal line really is talking about the timing of the divergence. So Drosophila as the outgroup diverged from the common ancestor of all chordates way back in deep time, back in the Ediacaran. Then we see the divergence of lancelets as invertebrate chordates from the common chordate ancestor during or right at the beginning of the Cambrian. So 542 million years ago marks the beginning of the Cambrian and of course the onset of the Cambrian explosion, that period of time where we see a huge diversification of the animal kingdom. So this shouldn't surprise us. And then we see at later points in time, the branch points that lead to the divergence of amphibians, reptiles, remember chickens are birds, also reptiles, and then divergence of mammals, and then divergence within mammals. So we are really commenting on when in geologic time the divergence points occurred. When did these species on these branches diverge from the common ancestor of that group? So yes, we can draw phylogenetic trees that do comment about genetic change, that do comment about the timing of the events. So making very strong statements about not being able to get that information from phylogenetic, excuse me, from phylogenetic trees now has to be taken a little bit more circumspectly. You can't be that definitive because it is possible now to include that data into a phylogenetic tree. But remember, unless it is specifically stated as part of the phylogenetic tree that that's what you are diagramming on the tree, you cannot assume that the horizontal lines are making comments. So if you go back and look at those other phylogenetic trees that we've looked at where there was no attempt, there were no time axes, there was no genetic change axis, there was no suggestion that there was anything other than strict relationship data being incorporated there, you cannot assume that the horizontal lines have any meaning. And that is true for most phylogenetic trees. The horizontal lines do not have any meaning unless that is specifically stated and specifically diagrammed and identified. So there are two other principles that we also need to comment on very quickly when it comes to drawing phylogenetic trees. Because part of the problem with starting to diagram data, and particularly molecular data, is that it's possible to draw more than one phylogenetic tree using the same data. This is why you can find different taxonomies and different phylogenies on the internet talking about the same group of organisms but being drawn by different people who view the data and diagram the data in different ways. But the two principles are known as maximum parsimony and maximum likelihood. Parsimony refers to the fact that the simplest explanation is almost always the best explanation. So if you've got a complex explanation of the phylogeny and you've got a simple explanation of the phylogeny, our experience tells us that the simple explanation is almost always the best explanation. Maximum likelihood says, and these kind of sound the same, but they're not. Maximum likelihood says that when you're looking at the phylogenetic trees that are documenting genetic changes, the tree that involves the fewest number of changes to account for the relationships is the tree that most likely occurred or represents the changes that most likely occurred. Why do we say that? By virtue of the fact that mutations are very rare, and even rarer are mutations that have an effect on phylogeny. So in terms of a phylogenetic tree that explains the phylogeny of organisms built out of genetic change data or genetic comparison data, it is most likely that the phylogenetic tree that requires the fewest genetic changes is the series of steps that most likely happen. So while parsimony and likelihood sound very similar, they are actually quite different in terms of what they're saying. So here are some phylogenies that are going to be built, in this case, on 3 species of birds. Now, what is ironic here is that the current edition of the textbook is talking about 3 different species of beetle, and you can take a look at the diagram and you'll see that. But if you do comparisons between the data that I've got here in this PowerPoint and the data that's in the textbook, it is in fact exactly the same data. It's just that the artist has drawn diagrams of birds here, and in the book, the new diagrams have incorporated pictures of beetles. It's exactly the same numbers, however. Even the genetic change table is exactly the same. So here are our 3 species of birds. We're going to draw 3 phylogenetic trees, but remember these phylogenetic trees are always and should always be considered to be hypotheses, because we don't know for certain which of the phylogenetic trees represents what happened. And even after applying parsimony and likelihood and picking one of the phylogenetic trees as the one that seems to have the greatest support, it's still just a hypothesis, because unless you were there to watch the changes happening, you have no evidence other than the genetic data and attaching it to the phylogenetic trees. So here's what you do. Here is our sequence data, looking at 4 different locations within a particular gene that is shared by all 3 species. This is the ancestral sequence, because remember, if we're defining a clade, we also include the common ancestor. If we're lucky, we may have a way of accessing what the ancestral DNA sequence may have looked like. based on sequence comparisons. So here is our data. If we look only at site 1, what we find is that the ancestral species and species 3 share a common nucleotide. So when we do that, we put the change not on the branch leading to 3, but on the branch leading to 1 and 2. So in the case of this phylogenetic tree, we put that change right there. It's a change from an A to a C. On tree 2, because 3 and 1 share a common ancestry, that requires 2 mutations, one on the branch leading to 2 and one on the branch leading to 1. Again, the branch leading to 3 remains unchanged. On tree 3, again, we need to apply 2 changes, one on the branch leading to 1, one on the branch leading to 2, and the branch leading 3 remains unchanged. Then we take a look at site 2. We see again that species 3 and the ancestor share a common nucleotide. At species 1 and 2 also share a common nucleotide. So there must have been either a single mutation that led to them on tree number 1, or there must be a multiple changes that led to species 2 and species 1 and species 2, with 3 remaining unchanged as a consequence of this second mutation. And we go through and we put marks on the various branches representing the different changes that have occurred. Then we take a look at the 3 phylogenetic trees and we ask ourselves, which is the simplest explanation and which requires the fewest changes? And immediately, tree number 1 stands out because it is a simple explanation. 1 and 2, based on the sequence analysis, share 2 of the 4 nucleotides, and species 3 and the ancestral species share 2 of the 4 nucleotide changes. And so that suggests that 3 is more basal to the tree than 1 and 2. And actually 3 is more basal to both 1 and 2. And so this is a simpler explanation. And it also results in the fewest genetic changes. So it also is in line with maximum likelihood. The phylogenetic tree that requires the fewest genetic changes is the most likely sequence of events that led to these relationships. And so that's why we would pick phylogenetic tree number 1 as the phylogenetic tree that is supported by the genetic data and accounts for the relationships that we see between these 2 species that we are defining as a clade. But it's still just a hypothesis. And it will be nothing more than a hypothesis. And if we get more data, if this is based on a single gene sequence, one gene is not a lot of evidence on which to build a phylogenetic tree. But it's basically all we've been able to do up until very recently. But now with the available of sequencing machines and large-scale sequencing labs, it is almost certain that we'll be able to take a look at a number of gene sequences and be able to create even more, let's say, quote unquote, accurate or more supportable hypotheses, let's put it that way, more supportable hypotheses on the basis of more data to establish which of the phylogenetic trees would be, would receive the greatest support by the data. But maximum likelihood and parsimony don't always provide us with the best explanation. So if we take a look at these, this data, which basically looks at gene sequence data or gene sequence differences between the 3 multicellular eukaryotic kingdoms. So animals represented by humans, fungi represented by mushrooms, and plants represented by tulips. These are the known sequence differences between the DNA of those three kingdoms on a gross analysis. And if we're going to draw phylogenetic trees, those trees need to take into account those differences, but also, ideally, the lines should end at the same point in time because all of the species represented are extant, not extinct. And so all of these kingdoms are present. And so there are two possible ways of drawing out the trees on the basis of sequence differences. So we need to add together percentages as represented by the horizontal lines to match the known percentage differences. So we know that the differences between a mushroom and a human is 30%. So if we look at this line, it's 15% plus 10%, that's 25% plus 5%, there's your 30%. For animals and plants, it's 40%. So if we've got a 5% and a 10% here, and we're assuming that animals and plants are more closely related than either are to fungi, which was the dogma in the past, then the only way that we can account for that is to have, again, the 10% difference that is known between the common ancestor to plants and animals and fungi. We need the 5% to fungi. We know that that's the change, but that means that there's got to be a 25% difference on the basis of plants as they've diverged from the common ancestor with animals. But that requires a huge number of changes. Remember, we're looking for a simple explanation. So a simpler explanation could be this alternative. What if animals and fungi are actually more closely related to each other than either are to plants? Something that was, up until recently, not thought to be the case. Again, the difference between plants and animals is 40%. So 20% plus 5% plus 15%, that's 20%. The difference between plants and fungi is also 40%. So 20% plus 5% plus 15%. The difference between fungi and animals is 30%. So 15% plus 15%. This actually takes into account the differences. The changes, the horizontal bars, are smaller, meaning fewer changes, a simpler explanation. This is the more likely explanation. This is the more likely phylogenetic tree. And in fact, other data is supportive of this. We now know that in fact, fungi and animals share a common ancestor with each other that is more recent than the common ancestor that they share in common with plants. Yeah, we are more closely related to mushrooms than we are to plants. Go figure. But again, the phylogenetic trees support this idea, and that's backed up by other evidence as well. So this is why this is an accepted relationship. So as I've said, phylogenetic trees are always hypotheses. We can't necessarily rely on them to give us the best explanation, but in some cases we can use them to find some interesting relationships. So if we assume that birds are descendants from dinosaurs, something that was actually suspected about 150 years ago when dinosaur fossils were first unearthed, but definitive fossils that clearly indicate that dinosaurs and birds are essentially the same group has not emerged until very recently, the 1970s and 1980s, with fossils that started to come out of a region in China called Liaoning. We may talk about this region again later in the semester when we get to the animal kingdom. We talk dinosaurs and birds. But it clearly indicates now, these fossils clearly indicate now, that dinosaurs had feathers. In some cases, they may have been completely covered in feathers. We do have fossils of creatures that are clearly dinosaurs in every sense of the word, our traditional dinosaurs, but they've got feathers on them. They're not birds because they have jaws and teeth. They don't have beaks. They're clearly unable to fly because they don't have flight feathers, and it's very easy to tell flight feathers from body covering feathers. And these animals do not have flight feathers, but they have feathers and their bodies are completely covered with them. And we now have a pretty logical explanation based on fossil evidence as to how birds probably arose from a group of dinosaurs called Saurischian dinosaurs, and specifically from a group of dinosaurs that includes velociraptors, the actual raptors. So birds are closely related to Velociraptor, the little villain in Jurassic Park. And we've changed our view of Velociraptor even. Jurassic Park got it wrong. Actually, the Jurassic Park movies got a lot wrong about dinosaurs and their origins. Fortunately, in some of the more recent Jurassic Park movies, they actually did correct some of the errors, but some of the foundational ideas for Jurassic Park are absolutely wrong. For instance, amber does not preserve DNA because of the nature of amber, it actually destroys DNA. That's one of the things that we know. The only types of fossilization that could actually preserve DNA are freezing and desiccation. All other types of fossilization would lead to the loss of the DNA very, very quickly. So amber, tar pits, mineral displacement, all of those lead to the loss of DNA. So realistically, the only way to extract DNA out of fossil remains was to have something that is desiccated, and there it would be fairly tough, or something that is frozen. And even there, it could be difficult depending on how badly damaged the cells are by freezing. So yeah, that's a story for another day, I think. Anyway, we even have some evidence that suggests that not just the Saurischian dinosaurs had feathers, but even the Ornithischian dinosaurs probably had feathers. There are a couple of fossils of dinosaurs that are clearly Ornithischian, but have feathers, something that is pretty recent, in fact, within the last 10 years. So feathers may have been uniform amongst the dinosaurs, and certainly they are features that are found in birds. So we can use birds as a closely related group, as a sister clade to the dinosaurs. In fact, probably as dinosaurs. A lot of paleontologists now say birds are not sister dinosaurs. They are sisters clades to dinosaurs. They are dinosaurs. So if you have chicken for one of your meals in the near future, or if you've had chicken for one, or any poultry, you've enjoyed some dinosaur. Crocodilians, as another group of extant reptiles, are distantly related to the dinosaurs. So what we have here is a phylogenetic tree that has extant species that bracket the extinct species, or the extinct group, the dinosaurs. So what we can do is do some cladistic analysis. Features that birds have that are unique to birds and not found in crocodilians must be derived features, and therefore logically found in all dinosaurs, because they're shared derived features. They would be defining features of dinosaurs. Features that are found in both birds and crocodilians would be considered to be primitive characteristics, because crocodilians would be an outgroup if we're defining dinosaurs as a clade that includes birds and all of those that are found in the fossil record. And so crocodilians would serve as a convenient outgroup and defined the primitive characteristics that would be in common to all reptiles that branch from the common ancestors of crocodilians, dinosaurs, and birds. So what sort of things are in common? Laying eggs, shelled eggs, nesting, migration, vocalization. These are all features that are found in common amongst crocodilians and birds. So those would probably likely be primitive features, which would mean that all dinosaurs would have these as a consequence of being primitive features, and they would show those. And we now know from the fossil record that that is in fact the case. Dinosaurs did lay eggs, they did nest, and in some cases they nested in large colonies, just like birds do today. They did migrate from one location to another. You may say crocodilians migrate. Yeah, just pay attention during the mating season for alligators. Alligators turn up in all sorts of locations, like pools in people's backyards. Those are the males. They're migrating to find a territory that they can call their own. So there is some migration involved in crocodilians. It is not as dramatic as we see in birds. So the sort of migration that we see in birds today is probably the sort of migration we would expect to see in dinosaurs, and as it turns out, that is in fact the case. Dinosaurs did migrate in large herds, and they did migrate from summer locations to winter locations and back again. All of this has been documented from the fossil record. So what are some features that are characteristics of birds that we might expect to be found in dinosaurs as shared characteristics that are not found in crocodilians? Well, obviously feathers are one. Another one that comes as somewhat of a surprise is the ability to maintain a high body temperature. So we know that all reptiles, modern reptiles, with the exception of birds, maintain a body temperature that is equivalent to their environment. That's why reptiles are constantly moving out into the sun and going back into the shade again. It's a way they regulate their body temperature. Birds maintain a high body temperature. In fact, their body temperature is higher than most mammals by doing the same thing that we do. They burn their food at very high rates and use some of the energy to maintain a higher than ambient body temperature. We also know that birds have hollow bones, something that crocodilians don't have. We know that birds have ancillary breathing organs to allow them to oxygenate their blood by virtue of the fact that flying is a very energy intensive process, and birds have one of the most efficient cardiopulmonary systems, in fact, likely the most efficient cardiopulmonary system of all extant species, and it's common across all birds, even non-flying birds. And so when we look in the fossil record, this is what we find in the Saurischian dinosaurs, which includes as diverse a range of dinosaurs as the velociraptors, the allosaurs, including T-Rex, the very large herbivorous dinosaurs like the Diplodocus, and the truly huge herbivorous dinosaurs known as titanosaurs. All of these have hollow bones, particularly the titanosaurs, and those hollow bones contain air sacs that assist in oxygenating blood by virtue of the fact that these animals were truly huge. They weighed 40 tons or more. They measured 100 feet from tip of snout to tip of tail. They had thigh bones that were over 8 feet tall and had a circumference measured in multiples of feet, diameters around 1.5 to 2 feet or more in diameter. These were truly monstrous animals. And a cardiopulmonary system that could fit inside that body simply would be incapable of oxygenating the blood and moving the blood rapidly enough to all parts of the body, including the brain and the muscles, to allow the animal to function as a highly mobile animal. And these were highly mobile animals. The trackways that they left behind indicate that they could walk at speeds up to 10 to 15 miles an hour. That is not slow. That is on the range. That actually is a little bit faster than a walking elephant. So these are not slow animals. These are quite mobile animals. And the only way they can do that is to thoroughly oxygenate the blood. And their cardiopulmonary system could not do that. But the hollow bones with the accessory sacs in the bones to help oxygenate the blood provide that solution. Birds inherited that hollow bones solution and adapted that to flight. They adapted the cardiopulmonary system to flight. Obviously, there are adaptations in birds that are unique to birds that are not found anywhere else. And so we can't necessarily extend some of those into birds, into dinosaurs. The fossil record doesn't support that. But it has forced us to rethink dinosaurs. What is a dinosaur? How did they behave? That is something that we've had to rethink on the basis of this phylogenetic tree that allows us to bracket the dinosaurs and do comparisons between birds as a sister group or as actual dinosaurs, and do comparisons against crocodilians, a closely related group as reptiles, and share a common ancestor with all the dinosaurs, but are not members of the clade dinosaur, which birds are. And so here is another practical use of a phylogenetic tree to gain more information about organisms that we can no longer examine in real time because they're extinct, but which allows us then to go looking for evidence in the fossil record and actually start to find the evidence that is supportive of the phylogenetic hypothesis. It's kind of a cool approach, but it means we've had to totally rethink what our ideas of dinosaurs. So here is a poor, unfortunate dinosaur that has been misnamed. Oviraptor refers to the fact that it was thought that this dinosaur was actually preying on the eggs in the nest of another dinosaur. It turns out, however, once a couple of other fossils were found, that the Oviraptor actually does not prey on the eggs of another species. It actually broods its eggs in the same way that birds do. The females probably, we don't know whether it's the females or the males, but at least one gender member of the mating pair actually broods the eggs by resting on them. Something that is uniquely bird in extant species. And they nest in large colonies. These colonies have been found, these nesting sites have been found in the Gobi Desert of all places. And there have been several large nesting sites discovered around the world now, which indicates that dinosaurs did in fact nest like their bird cousins, if you want, and nested in large mating colonies, just as birds do, a behavior that we would not be able to deduce in any other way other than recognizing the close relationship between birds and dinosaurs, making the assumption, correctly as it turns out, that birds really are dinosaurs in every term, in every sense of the term. In fact, most modern paleontologists will tell you now that there is no feature of birds that is not found in dinosaurs. There are no features in dinosaurs that are not found in birds. Birds are dinosaurs, period. So when we talk about the dinosaurs going extinct, most paleontologists that I'm aware of now say that it is the non-avian or the non-bird dinosaurs that went extinct. The avian dinosaurs, the birds, are still with us, or at least one line of birds, because as we may discuss a little bit later, there were actually 3 lines of birds, only one of which survived the end Cretaceous extinction. But more to come. Okay, So another break, and this time I will stop because we've been going for a while. So let me take a break here. So here's our concept check for this break. Would hair be a useful characteristic to define a clade of mammals within the larger clade formed by class Mammalia? Why or why not? So we know that class Mammalia is a clade. What we want to do is identify a clade of mammals, a subset of mammals within class Mammalia. Can we use hair as a defining feature or not? I'll see you after the break with the answer. So welcome back from the break. Let's take a look at the concept check question so we can move along from here. Remember that hair is actually a common feature of all mammals. So if we were trying to define a subset clade within the larger clade mammals, we would expect that hair would be found not only within the clade that we're defining, our subset clade, but would also be present in all mammals. And so we couldn't really use hair as a defining feature for our clade. It would have to be considered to be a shared primitive feature because the clade carries it, but all mammals carry it. So we'd have to look for other features as a defining feature within the clade that we're trying to establish. Hair would be present, but it's a primitive feature. It's not a shared derived feature because it's found amongst all mammals. So it's not a deriving feature, it's not a defining feature. So unfortunately, in this circumstance, we'd have to say, no, we can't use hair as a defining feature. So let's move on. So one of the reasons why we are really interested in DNA is because of what I mentioned back at the beginning of the presentation. If changes have occurred in species that result in speciation, all of those changes must at some point be reflected at the DNA level. And so we should find the evolutionary history of any species written as mutations in the DNA. The question is, when we start looking at mutations in the DNA, what type of genes do we want to use for the analysis? Because it turns out that if you allow some genes to mutate quite dramatically, that could result in the loss of activity. And if they happen to be really important genes for the continued existence of the organisms, that loss of activity could result in the loss of the species, extinction. So there are some common processes that are found across all of life and across all eukaryotes, across all mammals. So genes that have been used up until now, because of the limited ability to do sequencing, have been genes that have evolved either very, very slowly because they are central to the existence of the cell. So ribosomal RNA, and particularly small subunit ribosomal RNA, has been of a particular focus because all organisms require functional ribosomes, and there can only be a limited number of changes before the ribosomal RNA can no longer be functional. And we do know that the ribosomal RNA is in fact a ribozyme now. It is the actual enzyme that is doing the regulation of protein synthesis. And in the case of the large subunit RNA, it's the enzyme that is actually doing the linking together of the amino acids. It's the enzyme that forms the peptide bonds as the amino acids are linked together. So changing that RNA substantially will lead to loss of ribosome function. So these are genes that have evolved very slowly over time so that the function of the ribosome is retained. And that means we can use any sequence differences to look way back into deep time, going back millions and potentially billions of years into the past, because these RNA genes have been highly conserved. And so there will be a limited number of changes. And that gives us the opportunity to look for branch points in the deep distant past geologically. Mitochondrial DNA evolves more rapidly, but still you can't allow it to evolve too significantly, because mitochondrial DNA codes for proteins that are involved in cell respiration. And most of the complexes in cell respiration, particularly the mitochondrial complexes, involve at least one component that's coded in the mitochondrial DNA. So can changes occur? Yes. But so long as the enzyme function with respect to cell respiration is maintained, there is some scope for change. What we also know about mitochondrial DNA is the fact that mitochondria are inherited primarily maternally. There is some evidence now that suggests that sometimes mitochondria are also inherited paternally from the father, but predominantly mitochondria are inherited maternally via the egg. So there is some scope for mutation in the mitochondria, and this is why mitochondrial DNA has been very useful in tracking human migration patterns, and has led to the simple observation that humans as a species, Homo sapiens, originated in sub-Saharan Africa and migrated out of sub-Saharan Africa as recently as perhaps 60 to 80,000 years ago, but perhaps as early as 120 to 130,000 years ago. And it was only a limited number of individuals that migrated out, perhaps as many as 10,000, perhaps as few as 6,000. But the reason why we know that this is the case is because when we look at individuals in sub-Saharan Africa, there is a significant diversity in mitochondrial sequences. But as soon as we look outside of sub-Saharan Africa, we find that there is only a limited number of mitochondrial differences, mitochondrial mutations. But those mutations are significant, and they do allow us to map human migration patterns. And they have been used quite significantly to establish how Homo sapiens migrated, but also to some degree to look at migration patterns of those human species that are not Homo sapiens, like Homo neanderthalensis, Homo erectus, the Denisovans, that predated the appearance of Homo sapiens outside of sub-Saharan Africa, but show up in at least our nuclear DNA, depending on our ethnic origin, because of a small percentage of genes derived from those species, which indicates that obviously we mated with them when we encountered them as we migrated out of Africa. And mitochondrial DNA can be used as a very powerful tool to look at some of that as well. But in terms of looking at a broader selection of genes, what we need to recognize that there are two basic times under which a gene can undergo mutation without affecting loss of gene activity. And we identified these by two names, orthologous and paralogous. So orthologous genes are genes that are found as single copies in the genome and only have an opportunity to diverge from each other when speciation occurs. In other words, once the speciation event begins and two populations start to diverge from each other, there is an opportunity now for unique mutations to occur in the single copies of these orthologous genes, because they are now found in two different populations. And different mutations can occur in these genes as long as there's no loss of gene activity, as a consequence of the divergence that is leading to speciation. The other time under which genes can diverge and specifically in this case, without the loss of the original gene activity, and maybe even the possibility of accumulation of new gene activity, is in the case of paralogous genes, where for some reason a second copy or more copies of a gene occurs in the genome. So long as one of those copies retains the original function of the gene, the remaining copy or copies are free to mutate in another direction. And if you want a really good example of this, look no further than the fact that you see in a multitude of colors based on a red, blue, green, the RBG color system. Because if you look in other primates, and particularly if you look in monkeys, to whom we are very closely related as primates, they see in a two-color system. And the difference is the presence of a third color detecting protein, otherwise known as an opsin. Those monkeys that see in a two-color scheme only have two opsins, and therefore are limited in the number of colors that they can generate on the basis of their color receptors. We as humans have three color receptors. We have three opsins, and we know that one of the opsins, the most recent to occur, is actually a result of a duplication of one of the other two opsin genes that has then led to the second copy diverging a little bit through mutation from the original copy and taking on an ability to react to different wavelengths of light. It actually brings in red to our visual range and allows us to see red colors. And the advantage for that is when you think in terms of flowers and fruits that serve as good food resources, they change from green to red. If you can only see in a two-color system, you can't see the red, so you can't tell when the fruit has ripened. But if you can see red, now you can detect the changes in fruit, and you can determine when fruit is still green, and you can detect when the fruit is ripe and now ready to eat. And that has been a great advantage to humans and related apes that see in the RGB or RBG color scheme. We can see reds that other primates cannot see. And that came about because of paralogous genes, a second copy of one of the two existing opsin genes appearing in the genome and diverging through mutation to become a third color sensor protein. So this basically tells you, diagrams what I've been telling you about, what the situation is here. So this is the speciation event where a single copy of the gene exists in the ancestral species, a single copy exists in the two diverging populations, but now because they're diverging from each other, not only do the populations diverge, but the genes will start to diverge by accumulating unique mutations. And so these genes are a marker for a speciation event. These paralogous genes cannot tell us about speciation events, but they can tell us when unique features are accumulated within a species or within a group that share a common ancestry by virtue of the fact that we can see changes that have occurred in one of the two copies of the genes. One retains its original function and the second copy takes on a new role, and as I mentioned, with our color scheme. So one of the problems is that while orthologous genes are the ones that we want to look at as markers of speciation, we still need to be able to recognize them in the species that we believe are closely related to each other or are distantly related to each other. So finding examples of orthologous genes is not difficult to do because there are so many processes that we share in common and that are carried out almost entirely in the same fashion, using exactly the same enzymes, following the same metabolic pathways. Finding examples of orthologous genes amongst species is not that difficult. Being able to sequence them is, or has been up until now, hence the limited number of genes that have been used to create phylogenies on the basis of comparison of gene sequences. That is changing with the onset of gene sequencing labs and gene sequencing machines that can do the job quickly and easily and in large volumes, using, again, big data, computer algorithms to sort through lots and lots of small gene sequences to stitch them together on the basis of overlapping sequences to create an intact genome or an intact copy of a gene. Obviously, there's a lot more information that will be forthcoming as these new techniques are applied to genes that are of interest. So, quick concept check. We're not going to stop this time. We're going to keep moving. Define paralogous and orthologous genes. There should be a full stop there. Which of the two types would be most useful in establishing the phylogeny of a species? Explain why. So remember, paralogous genes are genes that exist within a species as multiple copies, 2 or more copies of the same gene. Orthologous genes exist as single copies in the species that contain them. When a single species splits as a result of a divergence, two populations diverge from each other by becoming reproductively isolated, those 2 single copies now also start to diverge from each other in their respective populations because of the accumulation of unique mutations in each of the 2 populations. Which would be most useful, as was established in the diagrams and in the notes. The orthologous genes are the ones, because those specifically are associated with speciation events. The orthologous genes only start to diverge from each other once the populations themselves have diverged from each other, allowing those genes now to accumulate their own unique series of mutations as the 2 populations remain reproductively isolated from each other. No gene flow, if you want to use the correct evolutionary terms. So let's move on. So the last piece of the puzzle in phylogenetics is the use of molecular clocks. Molecular clocks do allow us to get a window on divergence events that are not established in the fossil record, but they are based on some basic assumptions. They are based on assumptions that mutations occur at a fixed rate. So it basically relies on the uniformitarian idea, that the rate of mutation that we see today is the rate of mutation that has occurred in the past and is the rate of mutation that has occurred over time. It also relies on the observation that only certain types of mutations are permitted, particularly in the case of substitutions, and that those are the only mutations that have ever been permitted over the course of the history of life. It is also based on the assumption that the mutation of genes in 2 different species occur at the same rate, that the mutation of a particular gene in one species is occurring at the same rate as the mutation of the same species, the same gene in another species. So there is a number of assumptions that underline this notion of a molecular clock as a way of measuring divergence of new species or emergence of new species. We can use orthologous genes because they are the ones that start to diverge from each other at the speciation event or as a consequence of the speciation event. But we can also use paralogous genes as molecular clocks, not to identify speciation events, but to identify when duplication events occurred, and therefore when new features may have appeared as a consequence of duplication of genes and the divergence of those genes over time to give rise to a new function in the case of one of the two genes as it diverged from its duplicate. So we do create molecular clocks using both types of genes, but only orthologous genes can allow us to look at divergence of or speciation events in deep time. Paralogous genes allow us to look at when new features occurred within an existing species. So having said all of that, here is a molecular clock diagram. And it doesn't look anything like a clock, really, does it? It is basically looking at a particular gene that is conserved across mammals. And it looks at the number of mutations that have accumulated in the genes in different species, in different extant species, as a product or as correlated with the time that we know the species diverged from each other. And then a line of best fit, a linear regression analysis has been performed to create the line of best fit through the points. And you can see that by and large, all of the points fall on or near the line of best fit, indicating that there is a correlation between the known divergence time from the fossil record and the number of mutations. There is a linear correlation, except for those three pesky green points. And rather ironically, those three divergent green points are actually primate genes. For some reason, the rate of mutation in primates has not followed the same rate of mutation in other mammals. We don't know why, but obviously the mutations that have occurred in our particular copies of this gene have not occurred at the same rate. So that simple assumption that the rate of mutation in the same gene in different species occurs at the same rate, clearly this indicates that that may not be the case. So that would be a problem that we would have to take into account when we're looking at when we're using molecular clocks as a way of looking at divergence. One of the other problems is that many mutations are neutral. And if they have a neutral effect, they create genes that are themselves neutral and have no effect on natural selection and fitness, and therefore mutate at their own rate independent of their effect on the fitness of organisms. There's no selective pressure to prevent them from mutating at rates that may lie outside of the molecular clock. But by the same token, if they are free from natural selection, there is the potential that they may settle in to a regular rhythmic mutation rate, because their mutations are going to be the product of random events. And because they have no effect on fitness, there's no reason why a mutation that leads to loss of a particular gene function would have any effect on the fitness of the organism. And we have a bunch of neutral phenotypes, which you may be familiar with from Biology 1, things like a hitchhiker's thumb, attached versus free earlobes, hair on the back of your hand. There are all sorts of features. Those are considered to be neutral features, and they are the product of our genes. And the frequency of those features in a human population vary from generation to generation because there's no selective pressure for them. And so it's likely that these features have arisen not as the product of natural selection events, or at least not natural selection events that we can point a finger to. They may have been selected for in the past, we don't know, but we know that at the moment they are not subject to any evolutionary selective pressure. So the genes that are producing these features are free to mutate in one direction or another and to mutate at different rates. The assumption is, however, that that mutation rate is likely to settle into a regular rhythmic pattern. So neutral genes and neutral proteins may be the best selection for a molecular clock analysis. So I've covered some of these irregularities already. So the irregularities in natural selection, favoring some DNA changes over others, that's basically pointing to the fact that only certain mutations are permitted, only certain substitutions are permitted, other substitutions cannot occur or are not allowed to, or are subject to selection. If we are using a molecular clock, what we want to be able to do is not only to create the line of best fit, do the linear regression between mutations or numbers of mutations and divergent points. We want to be able to, or divergent points in geological time. We would like to calibrate that molecular clock against known divergent points in the fossil record. Unfortunately, prior to around 550 million years ago, the fossil record is pretty sparse. We don't have a lot of good fossils because we don't have a lot of hard parts to fossilize. And so the fossil record prior to around 550 million years ago provides a limited ability to be able to correlate the molecular clock. So even though we can make the assumptions that mutation rates have been fairly constant over time, it's difficult to establish that with certainty because of the sparseness of the fossil record, and also because we only have a limited number of points in the fossil record that we can use as calibration points. We have no way of being able to determine what goes on in terms of mutation rates in between the correlation points or outside of the correlation points. All we can say is that if there are changes in mutation rates in between our correlation points or outside of our correlation points, it appears that if mutation rates slow down and speed up at various points in time, the net effect is to give the appearance of a constant mutation rate. That's the best we can do. The other thing that is a limitation on molecular clock system is, again, the lack of a lot of gene sequences to use, because it was simply too difficult to sequence individual genes, let alone lots of genes. That will change. And that will change fairly rapidly, is my guess. But at the moment, we still only have a limited amount of information on which to base the molecular clocks. That being said, the molecular clocks are still quite useful, and we do use them, as you'll see. So here is a fairly simple molecular clock analysis, because it is a molecular clock in recent time. And so we can be fairly confident about the data that we're using. This is a molecular clock that looks for when the most common form of the HIV virus, what is referred to as HIV-1, the original HIV virus, entered the human population and would ultimately give rise to the HIV pandemic of the early 80s to mid 90s, when we finally started to get a new selection of drugs that would turn HIV from a pandemic, almost certain death sentence, to a treatable condition. The new sets of anti-HIV cocktail drugs are really good at controlling HIV to the point where they can push the titer, that is the concentration of HIV particles in the blood, to essentially zero, undetectable, using the most sensitive tests that we've got to detect the presence of viruses. And the good news in all of that is that if you can push the virus levels to that undetectable level, the chances of a person who is HIV positive either developing AIDS or passing the virus on to a partner through sexual contact is essentially zero. To all intents and purposes, is considered to be zero. So, when does the molecular clock indicate that the virus entered human populations? Well, if we do our line of regression, looking at the genome sequences of HIV, and HIV, by the way, is an RNA virus that uses an enzyme called reverse transcriptase to create a DNA copy of the RNA. That single-stranded DNA is then duplicated, and the duplicated DNA then inserts into the cell's genome. That's how the virus hides out in what are known as the CD4 cells of the immune system. So when we take a look at the known genomic sequences of HIV-1 and we do a regression analysis, you see that the line of best fit based on that regression analysis puts the period of time that the virus actually entered the human population in a high enough frequency that it would ultimately give rise to the pandemic once it had found populations that allowed for easy transmission from host to host. So that would be individuals who practice intravenous drug use using shared needles, people who initially practice homosexual sex, but ultimately, as would be demonstrated very, very quickly, anyone who practiced unsafe sex, whether you were homosexual or heterosexual, it didn't matter. One of the unfortunate parts of the HIV pandemic was that very early on, because it was discovered primarily in homosexual men, it became labeled as a homosexual condition. As we discovered very, very quickly, if you were heterosexual and practiced unsafe sex, you could very easily become HIV positive. The transmission could very easily occur. And likewise, using contaminated needles, so sharing needles for intravenous drug use, could result in the transmission of virus, because all that was involved and all that was necessary was coming in contact with contaminated body fluids, however that happened. Anyway, so it entered the human population in high enough numbers, according to the regression analysis, the molecular clock, around 1930, with an error bar indicating that it could have been as recently as the early 1940s or as early as perhaps 1910, 1912. This would have almost certainly occurred in West Africa, probably the area that we recognize as the Democratic Republic of Congo, or countries surrounding that area. Almost certainly came out of chimpanzees, because chimpanzees have a similar virus called simian immune deficiency virus, or SIV, which is itself the product of the fusion of two monkey viruses, most likely in the gut of a chimpanzee, or at least within the body of a chimpanzee that had eaten monkeys, which chimpanzees do, that carried these viruses, probably separately, so two separate monkeys, one containing each of the two viruses. When they were introduced into the chimpanzee that ate meat derived from both of them, that provided the opportunity for the two viruses to fuse together to give rise to the SIV, the simian immune deficiency virus in West Africa. It then enters humans as a product of eating bushmeat, which includes poorly cooked or preserved chimpanzee. And as a consequence, the virus then starts to be transmitted human to human. Why this period of time in West Africa? Because this was the period of great colonial development, when railroads were being put through to allow easy transport from the inner regions of Africa out to the coast. The rivers were also being used as transportation roadways, and that meant that individuals who lived in the interior of Africa, who consumed bushmeat or carried bushmeat to the markets to be sold, could do so more freely, which allowed for a more easy transmission of early forms of HIV from infected individuals into new hosts within the cities. And that allowed for the establishment of HIV in human populations. That would ultimately allow the virus to emerge out of colonial Africa into Europe and other locations, into America. Over time, and as the numbers of individuals that carried the HIV continued to expand, eventually it became a pandemic of all types of individuals. So this gives us an indication as to when HIV first got itself established in human populations that would lead to the pandemic. Did HIV enter human populations prior to this time? Almost certainly. There is some pathology evidence from samples that were obtained from individuals in the late 1800s and very early in the 1900s that give indications of the presence of the HIV virus. And you notice that that predates the time window that the molecular clock establishes. So it is almost certain that HIV entered human populations multiple times, but it was this window of time around the 1930s, so 1920 through 1940, that's the window of opportunity, that HIV finally got established in populations in Africa that had mobility that allowed them to come in contact with a broader range of individuals and allowed for the wider transmission of the virus rather than it being retained in individuals, in a limited number of individuals. It was now able to infect a wider number of hosts because of easy access to cities, easy access to larger populations. And once HIV got established in cities, then it would find easy ways to infect new hosts because of the close proximity and because of normal human behavior. I mean, let's face it, normal human behavior provides the opportunities for the viruses to move from host to host. And HIV has in fact adapted to living in human populations. When it first emerged in its pandemic form, it could be as little as 3 months from infection to death from AIDS for infected individuals. Before the cocktail drugs that we now use to control HIV appeared, HIV changed its behavior so that it could, well, let me back that up. That an individual who was HIV positive could live for 5 or 6 years or more before HIV re-emerged from the infected CD4 cells to collapse the immune system and trigger the onset of AIDS. So that's a change in behavior from HIV. It actually adapted to being present in the human population and became less lethal in terms of the time it took for AIDS to develop as a response to the HIV infection, to provide it with more opportunity to infect many, many more hosts, because it does not serve the virus any benefit to kill the host quickly. It can only spread from host to host so long as the infected host survives long enough to carry out transmission to a new host. And by becoming less virulent and not killing the host as quickly, it provides many, many more opportunities for the virus to infect new hosts. It is what is. So one more break, and again, I'm not going to stop because we are very close to the end. And my concept check question here is, what is a molecular clock? What assumptions are used to underpin the clock? So remember, a molecular clock is a correlation between rates of mutation in a gene and the time over which those mutations have occurred. To establish a positive correlation between the number of mutations and the time at which those mutations took place. To establish, or to allow us to use differences in genetic sequences between closely related or distantly related organisms, for that matter, as a means for establishing when the organisms diverged from each other or from their common ancestor, when we have no fossil evidence to establish that. What are the assumptions that underpin the clock? Well, the first is that the rate of mutations have not changed over time. The second is that the rate of mutations of a single gene found across multiple organisms is the same in all of the organisms, that there's no speeding up or slowing down on the basis of which organism the gene is located in. The assumption that neutral genes, which are not subject to natural selection or fitness selection, mutate at a constant rate because they are free to mutate and therefore are not subject to limited mutation. So these are some of the things that are assumptions that underpin the clock. There are probably a couple of others that I haven't mentioned at the moment that I can't recall right now, but these are the major assumptions that are involved in underpinning the clock and allowing us to use it as a way of measuring divergence points in geological time when we have no evidence from the fossil record. And you'll see that we actually do rely on this as we move through the rest of the semester. So finally, after thinking about all of this work that we do to create these phylogenetic trees, the use of cladistic analysis, the use of genetic information as the basis for forming genetic trees, what does that tell us about the tree of life? Can we actually create a universal tree of life, or is that something that is too simple to imagine on the basis of what we have learned by building phylogenetic trees using molecular data? Well, we do know that we can no longer support the 5 kingdom system. the kingdom of Monera is no longer supportable because we know there are two different types of prokaryotes, and Monera as a kingdom was defined as the kingdom of prokaryotes. Well, now that we know that there are two different types of prokaryotes, we can't have a single kingdom for them. And so kingdom Monera is no longer supportable. We also said the kingdom Protista is no longer supportable. Remember, it is very hard to imagine a common ancestor to all protists because of the huge diversity of protists. And we also have to bear in mind that while Protista was considered to be a kingdom of unicellular eukaryotes, many multicellular eukaryotes got placed in there because they simply could not be placed in the three multicellular eukaryotic kingdoms because they didn't fit. So given those challenges, it was decided to create three super kingdom classifications, and you recognize those, the three domains, the bacteria, archaea, and eukarya. And the genome sequences, as we have them right now, do support these as the basis for the origin of life, at least in taxonomic terms. So we don't believe that there is the need for another category that is super domain, a single category that brings all of these together in a single category. However, what we do when we draw up these three domains, and this is one way of doing it, and I'll show you another way in a moment, is define what is referred to as the common ancestor of all life, or to use a more, let me see if I can bring this up again, a more interesting term, Luca. Excuse me. Otherwise known as the last universal common ancestor. Luca. So what Luca was, we don't know, other than it was almost certainly prokaryotic, because prokaryotes dominated the landscape, both archaea and bacteria, and probably we think maybe bacteria a little bit earlier than archaea, but again, the jury is out on all of that. But almost certainly Luca was prokaryotic. Other than that, we can't give much more detail other than Luca was prokaryotic. But it was the last universal common ancestor. Another way of representing it, using the ribosomal RNA genes, is to look at it this way. So we have the three domains, domain eukarya, domain archaea, domain bacteria, and in fact, domain archaea probably contains at least one more clade in there. There is evidence for at least a fourth clade in domain archaea. But you notice that mitochondria and chloroplasts are now included in domain bacteria because of their origin as free-swimming bacteria. And so they are closely related. Are they organisms? No. But do they share some commonality with proteobacteria in the case of mitochondria and cyanobacteria in the case of chloroplasts? Yes, because they are derived from, in the case of chloroplasts, cyanobacteria, and in the case of mitochondria, probably beta proteobacteria. A clade within proteobacteria that is known to be aerobic and carry out cellular respiration in almost identical fashion to mitochondria. So those are inserted in there. And this is a relatively recent notion. This is probably the simplest way to represent the diversity of life on the basis of the domains. Those listings within the domains, so euglenozoans, forames, diatoms, and so forth, may or may not be kingdoms. This is one of the ongoing challenges of the three domain system. What do we do with all of these classifications. The only things that we know for certain is that plantae, fungi, and animalia, as the three multicellular eukaryotic domains, are still considered to be valid clades and are still recognized as valid kingdoms under the three-domain system. All other classifications, the jury is still out. Sometimes you see them listed as kingdoms, other times you see them listed as phyla, sometimes you see them listed without reference to where they sit in the taxonomic hierarchy. People are now less willing to assign them to some of those classifications simply because there's still a huge state of flux in our understanding of what the relationships are between these organisms. And I think that reflects something that I was talking about at the beginning of the presentation. As much as we want to pigeonhole everything, as much as we want to put things into organized groups, we have to remember that life does not necessarily recognize, in fact, I'll take it one step further and just say life does not recognize our categories. Even the category species, as I said at the beginning of the presentation, the line of demarcation between species is often very, very blurry. And in some cases, all that holds that line in place is geographic isolation, the fact that the species simply do not come in contact with each other. If they could, it's likely that they would collapse back into a single species again, because there's very little in terms of genetic difference to keep them apart. So it's one of the challenges of modern taxonomy, phylogeny, cladistics, to try and create categories that are meaningful in terms of what we know. This is our best attempt to date. Where it goes from here, that's a really good guess. No one really knows. And here's the fundamental problem that lies behind all of that. The problem is that in the course of time, there has been a process called horizontal gene transfer. Vertical gene transfer is from species to species within a clade or within a domain. But what we know is that there has been this cross-pollination, so to speak, cross-transfer of genes between different lineages within a domain and even between different domains. One of the problems with trying to explain the origin of eukarya is that they share gene sequences with both archaea and bacteria. And that is a reflection of the fact that these domains as prokaryotes are notorious for sharing DNA backwards and forwards. We know that this is the case between bacteria. We know that this is the case between archaea, and there is evidence to suggest that it has also occurred between bacteria and archaea as separate domains, because the evidence is found in our own genomes, but it's also found in extant archaea and bacteria. So there is probably no simple explanation for the genetic origin of life, which means that there is no simple explanation for the origin of cellular life once we have cells. And you can see that the artist in this diagram has drawn not a single common ancestor, not a leuca, but a pool of cells, out of which has emerged through genetic recombination and sharing of gene sequences, which almost certainly occurred during the early pool of prokaryotes, eventually the three domains as we recognize them today. Lots of swapping of genetic material backwards and forwards that would eventually settle out as the three domains as we recognize them today. And that's what leads to the difficulty in trying to establish what the common ancestry of all life actually is. It still remains an unanswered question, and I suspect will always remain an unanswered question. So one more concept check, and then we're done. Why is the kingdom Monera no longer valid? For the very simple reason, as I was explaining a little while ago, that the kingdom Monera was established to encapsulate all known prokaryotes. In 1973, Carl Woese, a biochemist in California, identified the existence of non-bacterial prokaryotes, which he originally named as archaebacteria, reflecting the fact that these organisms lived in environments that were very extreme and lay outside of the scope of what bacteria, as they were recognized, would be able to tolerate. This would eventually lead to the recognition that you can't pigeonhole all prokaryotes into a single kingdom, because they share enough genetic difference that they obviously share different common ancestors. And so when the phylocode became established as the set of rules for doing phylogenetics and cladistic analysis, it became very obvious that the prokaryotes needed to be split into two different groups. And as it also became necessary to accommodate all eukaryotes, because the kingdom Protista could no longer be considered to be valid either, it was decided to create the 3 superkingdom domains because of the diversity of bacteria and the growing recognition of the diversity of archaea. Each of those were considered to be genetically and biochemically and biomass-wise distinctly different enough to warrant their own separate domains. So we have domain archaea and domain bacteria, and then all eukaryotes could then be very conveniently placed in a single domain eukarya. So that's the reason why we no longer have kingdom Monera. So fortunately, Zoom cooperated today. We now have a presentation that is broken up into sections. I will post this as soon as Zoom says it's ready to go. In the meantime, if you have any questions, comments, or concerns about anything that I've mentioned here this afternoon, please feel free to contact me via email, or better still, come to office hours on campus or my virtual office hours. Check the syllabus for times and locations and how to access office hours. That's all for now. I will see you again back here next time when we start to talk about the wonderful world of prokaryotes, and specifically the very unusual world of bacteria. They are everywhere. See you again. Be safe.